Hello class, this is Miss CHEE. We're going to discuss the results of Lab 20 that we did today in lab, starting on page 101. Number 1, how are the appearances of the monocot and dicot leaf different? So we're talking about leaves here. So the primary difference is going to be that the venation is different. Here it is spelled for you. In the monocots it's parallel and it's webbed or network-like. I like that, network-like in the dicot. Number 2, how do the vascular bundles of the monocot relate to its vein patterns? Remember we talked about that with the leaves. Since the vascular bundles are housed in the veins, the pattern will be the same. So well, they don't really tell you. So in the monocot, it's going to be parallel, and in the dicot, it's going to be webbed or network-like. Number 3, how do the vascular bundles of the dicot relate to the pattern? Well, it's the same answer. The vascular bundles are housed in the veins, so the pattern is the same. So in a monocot, they're going to be parallel, and in a dicot, they're going to be webbed or networked. Number 4, how does the placement of the vascular bundles identify the stem as being monocot or dicot? So now we're talking about stems. The vascular bundles are scattered randomly in a monocot, but circular around the edge in a dicot. So if you're looking at stems, keep in mind we're looking at these pictures here. Remember the monocot and the dicot stems. Number 5, describe any differences you observe between the monocot and dicot stems. Well, again, the difference is where the vascular bundles are. They're scattered or random in the monocot. In the roots of the monocot, they're saying it's a ring arrangement. And they're arranged in a ring in a dicot. So the stems you have scattered in the monocot and ring in the dicot. That's page 101. And again, on page 98 is where we see these patterns, because that's where we have our pictures of our stems. And of course, that's what we looked at under the microscope today. Page 102 and 103, these are what we drew, a small cross section. This was their rendition of the monocot, and this was their rendition of the dicot. Ours really looked more like the pictures on page 98, I thought. And then number 7, how do the vascular bundles in the root of a monocot, so we're in the roots, look different than those of a dicot? Well, the primary difference in a dicot and monocot root is that vascular bundles are arranged in a ring for the monocot roots. Okay, not like the scattered of the stem, but in a ring. And the xylem forms an X in a dicot ring. Well, we didn't really see that, so I'm going to show you what that means. On page 96 is where our roots were. Okay, so you remember, here's our monocot root or our tap root, and the xylem and phloem are in here, okay, in the ring. And in the dicot root, the xylem and phloem are in the center here. And what they're talking about is, here's another rendition. This is the same rendition of this. Okay, but it's a different type of drawing. I got it off the internet. And if you look down, here it is, and here's the center where the vascular bundles are. And this is what they look like, an X at higher magnification. So on our book, though, we couldn't see that, like our book picture here. And I don't remember viewing it in the microscope. It would be right in here. Okay, so that's what it means by there's a ring for monocot, and then the xylem forms an X in the dicot ring. Number 8, from your observation of the cross-section of the carrot, does the carrot look more like a dicot or monocot? Well, it's going to look like a monocot because it's a taproot. All right, your homework consists of reading the entirety of, what's the next lab? 21. Oh no, you're doing 21. That's the leaf lab. Performing the leaf lab and then reading the entirety of 22. Please don't forget to collect those worms and to email me if you have any. Have a great week.